Hello and welcome to this video on using fermentation to make protein. Typically, fermentation leads to ethanol and consequential drinking that causes weight gain, and not protein that is used to get swole. Typically speaking, a protein to get swole either comes from a can or an animal. Joking aside, fermenting to make protein is a fast-growing field of research that is being used to produce supplemental protein for things like dietary intake, animal feed, and possibly a very long time from now, in space-based protein production. At present, the technology is very limited in capacity and scope of production, and arguably the most advanced and reliable example is going to be casein. The casein is most frequently found in milk. This is a limited range of use and much more expensive and difficult to produce than milk from cows, sheep, goats, and so on. But that is a issue to do with the novelty of the technology, and not so much the actual idea itself. So if we're looking very distantly into the future, it's entirely possible that, although not Soylent Green, things like protein will be made in a similar manner. Not quite, but similar. There are a few different ways that this is done. There's either going to be precision fermentation, which is probably the most reliable and expected to achieve some modicum of success method, and a few others, but we'll focus mostly on that to start with. What we're doing with precision fermentation is essentially making something that's genetically engineered, generally something like yeast or some bacteria, to produce a very specific protein, hence the name, precision fermentation. You're precisely trying to make one particular thing, like casein, whey, in case of, say, egg proteins, oval albumin, ovomucoids, or similar, and in some cases a heme from meat. Although in other cases you might be saying myoglobin or similar. All of these are exactly the same as their, let's say, naturally occurring counterpart. That is what you'll get out of an egg, out of milk, out of any kind of meat product. It's all exactly the same, except where it comes from. And this does, to an extent, help considerably in use in, say, nutraceutical applications, or even food reinforcement. Most of the research at the moment is largely focusing on, let's say, high-value proteins, such as those used in dairy, like casein and whey, in animal-free products, for example, making yogurts, cheeses, milk, and so on. So there's a good reason to target those in terms of the, let's say, dairy industry. In terms of egg production, we've already mentioned the oval albumin and ovomucoids, and this is mostly targeting vegan baked goods and similar supplements, which is both a very small market and a market that should never be supported. But if we ignore the vegan issue for now, there's possibly uses for it. And then finally we have meat proteins, a heme being one of the more common examples, that is essentially the iron part of meat. This is both used to enhance flavor and color, but also, rather importantly, for giving people iron in their diet. And then finally we have collagen and gelatin, which is used in many foodstuffs, but most, let's say famously, gummy bears, and obviously desserts and similar. The reason this technology is so important is, well, let's use a simple example here. We've previously published a video all about how, with the Watney protocol, that is, you get stuck on Mars with a handful of potatoes and need to live for 2 years, you would starve and die before then. But if you had technology like this with precision fermentation, you would arguably have a better chance of surviving. Then again, remembering that this is relying heavily on fermentation, so it would take time, you need to have the necessary things to support the fermentation and so on. But under the right conditions, it's entirely feasible that it would work. Maybe. It does, let's say, build on a number of technological advances, primarily genetic engineering or editing tools. The CRISPR and Cas9 are the go-to options there, although other emerging technologies may or may not replace this over time. It also does make use of large language models and similar things to predict not only the products that will be generated from certain modifications, but to create the necessary models to improve any efficiencies in producing these. If that does work, and there's a big caveat to all of that, we'll get to in time, you could decrease reliance on much more, let's say, difficult to produce consumables. For example, feedstocks, sugars, CO2 in the atmosphere. You could instead use much more readily available, CO2 being readily available, but much more readily available products that aren't anywhere near as difficult to, let's say, process, ship, and do in other ways that isn't necessarily, let's say, convenient for, in particular, interstellar travel, which would be the ultimate goal. But certainly in the short term, it reduces cost to companies to use this technology. We'll now look at a few of the different techniques, let's call them, that are available. First, we'll start with the simple one. This is solid-state fermentation. This is taking lignocellulose feedstock, essentially things like straw and hay, and using that very high and abundant but cheap biomass via enzymes, converting it into something like a protein. This is perfectly effective. It works extremely well, and solid-state fermentations can increase the protein content in this by up to 161%. So that's a fairly significant increase, but understand that it's not entirely perfect, but it works very well. We then have gas fermentation, which is more complicated, but obviously gets slightly better results. But what we're doing here is looking at things in the air like carbon dioxide or methane, and using that along with feedstocks to convert that into protein as well. So it has uses in very particular applications, for example a space station where you have humans exhaling CO2, and that CO2 has to be either disposed of or somehow used. In this case, using it is actually quite a good option. Then we have the genetic engineering element. We've spoken about the fact that it's either going to be bacteria or fungi in the form of yeast for the most part, and there's a good reason for that. Microorganisms like Saccharomyces cerevisiae, yeast, and more accurately, mostly the beer brewing yeast, Piscia pastoris, also a kind of yeast, E. coli, which is a bacteria, and certain other species are used partly because of how well we know them. These species are very familiar to researchers, and we're not talking like Alabama family levels of familiar or stepsister stuck in a drying cabinet. No, we're talking very much about the down to the very basic genetic code familiar. And so we know what to do for the most part in terms of, say, promoter optimization. That is, taking certain genes that will cause production of certain other things, and to an extent, what we can do with those. If we, to use a simple example, use the AOX1 gene in the Pastorius, we can then get them to produce roughly 3.5 grams a litre of bovine lactoferrin. Of course, being completely familiar with the various pathways involved is why we can do that, and why, to an extent, we can redirect, move, and otherwise tweak all of these cellular pathways and resources into protein production. It's not necessarily true that we can do that with everything. The next option we have is biomass fermentation. This is very much similar to what you do with, say, straw, hay, and similar, except rather than relying on these other outside sources and modifying them, you literally have something like, say, a microbial mass producing what you want directly. It's very much similar to how we get insulin now, where insulin, depending on the exact method used, is either made as a whole protein product or the various required components of the insulin, which are then assembled after that. This is better suited for use as a direct food supplement, as you can basically take what is, for all intents and purposes, the, let's call it a brew, much like with beer, and siphon off the liquid component, add more liquid in the form of the various resources the cells will need, and you have a continuously functioning and continuously producing source of protein. Or, alternatively, you can just use the microorganisms directly. So you have something very similar to, say, how a sourdough might work, where you siphon off half of the sourdough, which you then use, and you feed the other half to grow it up to size again. And this is arguably more likely to happen, simply because something like Saccharomyces cerevisiae is nearly 50% protein. You have microalgae, which can be up to 60% protein, and various other species would obviously have to be selected based on their high protein content, but also the fact that they need to grow fast, and be very efficient in use of whatever substrate you're going to throw into it. So cerevisiae works well because it's just sugar, and sugar is typically cheap as it can be sourced and used in many ways, whether talking about cornstarch, wheat, some sort of synthesized sugar base. There are many options, and the other examples you mentioned obviously are similar. The fermentation process, if you're going to do that, is fairly straightforward. It's in many respects similar to what an aerated bioreactor is. That is, for all intents and purposes, the same as a fermentation tank used in home brewing, or even brewing for a microbrewery. What you have is a giant vat, essentially. This vat is filled with the requisite ingredients, glucose, ammonia, various, let's say, waste-derived substrates, such as spent coffee grounds, peels from various things, and so on. And this is continuously aerated to provide oxygen. That's then kept at a relatively constant temperature and pH, amenable to whatever your microorganism is. So, for example, just under 30 degrees Celsius and a pH of about 6 for yeast. And So long as you provide the necessary ingredients, like glucose, ammonium, or nitrates in a broader sense, you can get up to 65% protein in up to a week. Arguably less protein in less time, but that's still a lot depending on the obvious volume of what you're doing. But that's with submerged fermentation. Solid state fermentation's a little bit different, but it's very similar to, say, if you've ever grown mushrooms, where you take something like wheat and straw, a rice bran, or even food waste, and you pack it into a very container that's suitable to your purposes. So, for example, mushrooms, it might be a large tub, and this is then fed with some water, but not an excessive amount. You then essentially wait for the microorganism you're using to work on this and modify it, and typically speaking, sort of exude the protein that it makes out of its body. It also makes less protein as a result, somewhere between 2 and 16% over the same rough week period. The gas fermentation is useful as it is a very high yielding process based on the current somewhat successful attempts, and it uses things like atmospheric CO2, methane, hydrogen, and so on. You do need certain bacteria, and in this case it is nearly always going to be bacteria rather than fungi that work with this. And what they can do is create up to 70% protein using it. 70% tends to be on the high end, with most, let's say, successful attempts sitting closer to 60-65%. Shifting to the biggest issue you're going to find is the substrate utilization and, to a lesser extent, waste from this. For example, a lot of what is used in this is actually not the primary ingredient you're going to want to be getting from something else. For example, they may be using waste byproducts. So, for example, molasses, whey, and lignocellulose residues are all byproducts of other industries. Whey from cheese making, molasses from producing sugars and similar, the lignocellulose from, say, making paper. All of these things can then be arguably used and yield a fairly high amount of protein for what they are. It is important to note that depending on exactly how you set this up and depending on how, let's say, direct and intentional the substrate you're using is, so therefore not necessarily a waste byproduct, you will get significantly more protein. If you're looking at being more efficient by using waste product, you do lose efficiency at 45% protein production, but at the same time, you are removing an entire unwanted compound or chemical, byproduct, etc. from your supply chain. And of course, this does look at things like carbon capture. Obviously, in space, when nobody hears you scream, they won't know that you're slowly dying from CO2 poisoning, unless of course you have this sort of system where you take that atmospheric CO2 and turn it into protein, and obviously now you just suffocate to death. And to process all of this mess you've made throughout the processes we've described so far, first of all you have something like centrifugation, and that is you let you just spin it until you separate the protein out from everything else. You then need to filter this because there will be things in there you don't want, and possibly have to wash it to remove any of the residual substrate, which may or may not be necessary. This is arguably not so complicated or difficult to do, but it is relatively resource intensive in that you will need a dedicated facility of sorts. You'll then need to take whatever it is that you've produced in terms of protein, and you will then need to an extent, desiccate or try this out to produce your protein, preferably in a powdered format, because that will then allow you to create a meat analog. You may also need to treat it with heat to inactivate any possible, say, microbes, bacteria, fungi, etc., that have somehow survived everything you have done so far. And finally, simply to make it palatable to humans, you would then need to, say, remove any heat. You would then need to shape it, and you would possibly need to add other things to it to make it more meat-like. Some research indicates that you are actually better off combining different kinds of microorganisms, mostly fungi and bacteria, together, not only to improve the protein yield, but to improve the nutrition overall. One of the major things that will be a problem with all the research we've mentioned so far is that if you are going to take this and you apply it in something like, say, a space colony, you need to be very mindful of the fact that it has, well, it's a very oxygen-intensive process. So not only are you using a lot of carbon dioxide, which is good, you're also likely to be using a lot of oxygen. And so you have both a net loss in the negative aspect of what you breathe out, but also a net loss in what you need to breathe in. So in order to get optimal biomass growth, you need to balance these two factors very carefully. The only true issue with that in most terms is that it applies almost explicitly to essentially gas-based versions of this sort of system, whereas a solid state and similar, not so much, but still, it's important to be aware of it. Next we have the possibility of, say, bioactive peptide production, which is very much similar to what we mentioned earlier about the idea of, say, insulin. This is when you take bioactive peptides that are found in protein-rich substrates, and functionally you use them as the supplement. What you're doing is, say, taking milk, soy, or whatever else it is, and by using microbes of various kinds, and particularly the proteases, you can break them down and turn them into what you want, that being the protein, leaving the rest of it mostly intact. This is good if you're looking at making drugs in particular, things like ACE inhibitors, for things like, say, hypertension. The process is mostly the same as what we've already mentioned. It's a little bit different, but largely similar. So, for example, you take your fungi, so Aspergillus oryzae, the same sort of thing, used in making sake, and they release proteolytic enzymes, these enzymes that will break down proteins. But you need to lower the pH arguably even further, somewhere between roughly 4.5 and 6.5, and elevate the temperature above 30 degrees. Arguably no higher than 45, although this will vary somewhat. The benefit of this somewhat more niche situation is that it is much quicker. It works in between 12 and 48 hours. And so this gives you a substantial amount of, arguably, very useful peptides. Peptides being the things that build up larger proteins. It's useful if you already have a plant-based source of protein that you don't have ready access to, or you want to convert into something arguably more easily transported and used. Much like everything else, you want to centrifuge or centrifugate, you want to filtrate, ideally through ultrafiltration methods, and you want to perform separation using the bioactive peptides as the targeted thing you're after. You then may or may not need to, necessarily, try and identify specifically what you've gotten out of this whole process and separate it out into those discrete categories. So we mentioned the role of, say, making an ACE inhibitor, but you might be looking at, say, antimicrobials instead, which would make sense. In space, and for that matter, in certain limited situations, you may need to be completely self-dependent on any kind of medication. To optimize all of this, though, you do basically need to make sure that everything is done exactly as it should be under exactly the right conditions, especially since under some of these circumstances, you are more than less trying to make a medication. You don't want to get the medication wrong. That is how you get sepsis and die, which is already a high risk in space. You do get a, admittedly, very low yield, at between 1 and 2 grams a litre, but 1 or 2 grams is a lot when you consider the relative, let's say, functionality of what you're doing. You don't necessarily need a lot. Now for more depths on the gas methods that are available. First of all, we have gas-fed bioreactors. These are what take those atmospheric CO2, methane, hydrogen, and similar, and use that as the carbon source. This uses, say, species like Methylcoccus, and they oxidize the methane, which then produces a very large amount of biomass in terms of protein, up to 70%, which is a lot for what it is, remembering. You're putting in 70%, say, for a kilogram of this particular gas, CO2 mostly, and out of that you get 700 grams of protein. Just for the sake of imperial units, it's roughly 2 pounds, a bit over that, of biomass going in, and out of that you get about 1.5 pounds, thereabouts, of protein. It's arguably somewhat more efficient than the other options, but at the same time, although it's faster and it is more efficient, it does obviously require considerably more skills to match it. It does also require significantly more modification of the microorganisms you are going to be using. So it's a catch-22 scenario where it is better, but it's also worse, so choose your evil. In order to make sure this all works well, you basically need the same sort of considerations as before. That is, make sure you're monitoring it well, pH issues, a dissolved gases issue, the amount of biomass concentration to ensure that everything is running optimally. You also want to be analyzing, typically speaking here, using near-infrared spectroscopy rather than, say, HPLC, to figure out what it is you're getting exactly. Now, you can also use HPLC, but it's not necessarily absolutely required. And then finally, you want to analyze the exact bioactivity of what you've pulled out if you're not looking at just a basic protein. In order to run all of this, we've sort of spoken about the, let's say, large-scale systems. That is, if you were to just do this and what it is that it is, fundamentally what distinguishes one from the other. But they all do have the same fundamental system. It is a bioreactor. Now we'll use a submerged fermentation bioreactor to use a simple example for you. This again goes back to something vaguely similar to a brewing environment. You have your large stainless steel tub, the bioreactor. This is anywhere between 50 litres all the way up to, say, 300 litres. And this is a surprisingly standard size. You then have the pH, which again is anywhere between 4.5 and arguably up to 7.5. The temperatures can be as low as 25 degrees Celsius and, depending on what you're doing, up to 45 degrees Celsius. What is distinct and very different from, say, home brewing is how much oxygen is dissolved. Up to 50% in some cases, and relatively constant agitation. So it may be as little as 20% oxygen and 50% is on the high end. Agitation, however, is relatively constant, but again, it ranges between 200 and 800 rotations per minute. You're then going to add your various substrates, glucose, sucrose, glycerol for carbon, and you may even have other cheaper options, for example, lignocellulose from the paper industry, and in the case of gas fermentation, CO2 from the atmosphere. You then take this and you can then derive your product. So if we're talking about, say, a 50-litre tank, 70% of a 50-litre tank is still 35 litres of protein, and of that you can get between 1 and 7-ish grams per litre, which again, we're not talking about a shattering amount here, but this is also very early days. Also, this is going to be feasible every 48 to 72 hours from memory. Of course, that assumes that you just do this once, produce your results, and that's the end of it. But you can also do a continuous production method. Things we mentioned earlier about the idea of you basically siphon off a certain amount of the substrate, and you leave whatever you're using to produce the protein present, and give it more substrate to work on. If you are looking at using this though to produce a useful product, unlike a lot of other methods where you do have a relatively consistent loss, that is, for example, if you're using meat, well, you have to account for bones, connective tissues, organs, and so on. With this, it's almost a static loss, admittedly static in that you know it's going to be between these two measures, between 10 and 20% of the protein you're producing, making it a loss, which could be improved via optimization. Optimization, of course, machine learning is the major way there, to not just figure out how to conduct the fermentation, but what modifications to make, what processes to alter during production, or more accurately drying and storage to create a, let's say, a useful protein product, and so on. So it's not that we're not yet able to minimize those losses, but that those losses are, at least at this point, what they are. It will change over time. And this brings us finally to the not only challenges, but arguably future directions in this kind of research. The scalability is the major problem at the moment. Most of the fermentation infrastructure needed at the moment is incredibly expensive, and, to a lesser extent, it needs a lot of hurdles to be jumped over before it can get approval to be sold as a product. Consider how long it has taken for some of the meatless meat products to reach market because of those regulatory hurdles. So you have huge investment up front by companies who may not see a return for years in some cases and possibly decades in others, especially for novel foodstuffs. Some countries also have massive problems around genetically modified foods, the European Union being a perfect example. And, well, consumers. Most people are not going to look at, say, their bizarre protein product and go, yeah, I'm not going to touch that. They're probably going to think, okay, is it cheaper? And if it's not, I'm going to stick with conventional stuff. And even if it is cheaper, they're going to look at it and go, well, I know what, say, soy protein is, and I'm happy to go with that. But this bizarre bacteria-derived thing, no thank you. Genetically modified food has a massive PR issue in that respect. Future research would obviously be focusing on things like making it more cost effective, improving yields, improving the final product. You don't want to be eating something that looks like congealed mess. You would rather consume something that has at least a vague semblance to what it is you're wanting to consume. So basically make it look like animal-derived protein even if it's not, which is something of an irony and something of hypocrisy. It's like making bacon that's vegan. That's not bacon anymore. It's just weird congealed mess that you formed into a mud pie that resembles bacon. But assuming that this is not being done for such reasons, it would make sense to make it more viable to sell and certainly more palatable to those who are going to consume it. And finally, just simply cost issues. As we mentioned, even if it is cheaper, people may not buy this, and if it is cheaper, it depends on who will be buying it and is it actually a competitive product. The methods for fermenting protein involve a lot of different very complex methods, techniques, and facilities that have all been tailored to specific attempts to achieve something. Whether you're trying to be precise in what you're doing, whether you're using biomass, whether you're after bioactive peptides, or anything else. And it relies heavily on the skill set and knowledge base put into it to create whatever it is that you're trying to do. For example, genetically engineering microbial organisms. They're trying to design a reactor that's going to work for all of this. The methods used to optimize what is happening, whether or not they're going to produce a broader type of protein, so let's say a whole group of them, or if you only want to derive a single kind of protein out of what it is you're using as a substrate. On the other hand, because we are largely talking about the idea of using biomass and mostly microbial organisms, you're able to use a wide range of substrates, and you're able to minimize any possible waste products that come out of this or waste products from other processes that would then be used in this. This is what should create a relatively diverse and relatively easily leveraged technology that allows for the production of not just protein in terms of consumables like food or supplements, but also many other different kinds of products. For example, pharmaceuticals in the form of antibiotics, or possibly just basic research things like antibiotics that are used to, say, conduct antibiotic testing. Things like this allow for very efficient, fairly flexible, and to a certain extent, let's say, customizable production of various things. But at the moment, it's not necessarily scalable, and it's really only suited, at present, for making something like supplemental protein that's used for getting swole, or being a gym douche. But either way, the results are much the same. It's not yet there for a large-scale production of anything other than dietary supplements. It's still pretty damn cool. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. And please do post any comments, questions, or suggestions that you have below.